So it's my great pleasure to welcome Jodi Flaws for our seminar today. So Jodi is a professor of comparative bioscience at University of Illinois, Urbana-Champaign. So she received her bachelor's in biology from St. Xavier University, a master's in biology from Loyola University of Chicago, and then completed her PhD in physiology and toxicology from the University of Arizona. Following her degree, she then completed postdoc positions at Johns Hopkins and also at University of Maryland before taking a position as an assistant professor at the University of Maryland, where she was subsequently promoted to associate professor. In 2006, she accepted a position as professor of comparative biosciences at University of Illinois Urbana-Champaign. And there, her research focuses on determining the mechanisms by which environmental chemicals affect the ovary and female reproductive health. So her research is funded by grants from NIEHS primarily. She's funded, among others, for a summer undergraduate research experience toxicology program. There's a T32, 2 R21s, 2 R01s, I'm sure some others. She's published over 300 peer-reviewed publications, currently serves as secretary for the Society of the Study of Reproduction, as well as an associate editor for toxicological sciences. She's received numerous awards for mentoring, research, and service, including just a few. University of Maryland Student Mentoring Award, Patricia Sokolov Outstanding Mentor Award, Dr. Gordon and Miss Helen Kruger Research Excellence Award, etc. I can certainly attest the mentoring awards are well deserved. I know several of Jodi's trainees who have turned into fantastic researchers themselves, and have heard nothing but just warmth of Jodi's mentoring style. And she is not only a very supportive and giving mentor to her own lab group, but also to early career researchers in general, which I have experienced. So it's truly been a pleasure to get to know her over the last few years, I've been years now. But she's also doing just incredibly cool science that she's going to be able to tell you a bit about today. And so I'll hand things off to her for that. Thank you, Jodi.
Thank you so much, Chris, for that introduction. And thank you so much for inviting me. Chris is truly a superstar in our field, and it's really nice to be here and see how well he's doing. And a special thanks to Lynn for organizing all of my visit. I know it's a lot of work to do this, and my visit has been perfect. I've really enjoyed everything, so thank you. Can everybody hear me okay if I don't use the microphone? So what I want to talk about today are some of our work on environmental endocrine disruptors and female reproductive aging. Oops. I'm already failing this. Yeah. Okay. I'm going to put a pointer on the screen because I think this one's not going to show up. Okay, I might need help with that. Okay, sure. Because this one's not showing text. Change the color. Spotlight.
There you go.
There you go. Okay, thank you.
Nice. Well done.
Oh, I'm in trouble. Sorry.
In your upper left now.
Oh, thanks. Do I just need to leave that bar there?
You might be able to move it to the left.
Didn't like that.
That's fine.
All right.
Okay, all right, sorry. Okay, so what I want to do is give some background, and I know that not everybody in the audience is a female reproductive biologist, so I want to give enough background on female reproductive biology and aging. I'm going to talk primarily about early onset of reproductive aging And then the chemicals that we're most interested in my lab are phthalates. So I want to give some background on that. And then I'll share with you some of the results from our studies on the effects of phthalates on reproductive aging in mice and women. And I'm going to be sharing some data that are really kind of new to us and we haven't actually published yet. So hopefully I can get some good feedback on that. I'll share our hypotheses, experimental design, our results, and our conclusions. Okay, so female reproductive aging is really the transition of a reproductive state to senescence in animal species. In women, we refer to this as menopause. In non-primates, we will call it reproductive senescence. Now, in women, this usually occurs around 50 to 51 years of age on average, and it's characterized by low estrogen levels, menstrual acyclicity, and loss of fertility. One really interesting thing about the female reproductive system is that it tends to age faster than other physiological systems. And in fact, it's considered a sensitive indicator of aging in general. And reproductive aging is a public health concern because it's associated with increased risk of chronic diseases and conditions such as osteoporosis, depression, cardiovascular disease, and infertility. All of these diseases and conditions, the risk for them goes way up when a woman becomes postmenopausal or has transitioned from a reproductive to a non-reproductive state. Now, early reproductive senescence, so earlier age at menopause, even by a few years, is even of greater public health concern. The reason for this is that women will experience a greater risk of infertility at an early age. And this is a problem for women who delay childbirth for professional and personal reasons. So for example, most women, fertility is starting to decline in the early 30s. And if you have an early onset of reproductive senescence that can be really early 30s or even late 20s, and you wait to have children until that time, you're going to have more of a difficulty if you've got an early onset of senescence. And early reproductive senescence even causes a greater risk of chronic diseases and conditions leading to early death compared to women who have a normal age at reproductive senescence. So women that have early reproductive senescence, they have a greater risk of cardiovascular disease, osteoporosis, neurological problems, as well as infertility. And the risk of these sometimes goes up 2 to 3-fold in early menopause or early reproductive aging as compared to a normal age. So I want to give some background on how normal reproductive aging occurs, because this is happening in the absence of even chemical or any other environmental exposures. So in normal reproductive life, in the brain, there's a structure called the hypothalamus. The hypothalamus will synthesize and secrete a hormone called gonadotropin-releasing hormone, or GnRH. This GnRH then can act at the anterior pituitary and cause it to secrete follicle-stimulating hormone, or FSH, and luteinizing hormone, or LH. Both of these hormones go to the ovaries, and they do two things. One, they stimulate growth of the follicles, which contain the egg or the oocyte, and they also stimulate production of hormones. Some of the hormones that the ovary makes are inhibin, which is a protein hormone, and the sex steroids, estrogen and progesterone. Now, when levels of inhibin, progesterone, and estrogen become high, inhibin can negatively feed back to the pituitary to tell it to quit making FSH. Estrogen and progesterone can negatively feed back to the pituitary, tell it to quit making FSH and LH, or it can feed back to the hypothalamus to tell it to quit making GnRH. So it's kind of like a thermostat system. Now, what happens during reproductive aging, I'm going to start from an ovary-centric view because I study the ovary. But in the ovary, what happens is those follicles that are there, female mammals are born with a finite number, and they naturally die off over time. This is why women, when they hit menopause, they lose fertility because these follicles have decreased over time. So one of the first things that happens in normal aging is the follicle numbers will decrease. And because follicle numbers decrease, you not only have not enough oocytes for ovulation, but but levels of inhibin, estrogen, and progesterone go down because follicles are secreting those hormones. Now, when inhibin, estrogen, and progesterone go down, there's not enough feedback to exert negative feedback to the pituitary and the hypothalamus. So the consequence is there's nothing to shut off the hypothalamus, and it continues to make GnRH at high levels. And those high levels of GnRH stimulate the pituitary to make high levels of FSH and LH. And actually, these levels of FSH and LH become super high, and they're actually toxic to the ovary, and they cause even faster loss of ovarian follicles over time. So during reproductive aging in mammals, what we expect to see are a decrease in follicle numbers, high levels of LH and FSH, high levels of GnRH, low levels of inhibin, and sex steroid hormones. So because of the physiological changes that happen during reproductive aging, there are several markers of reproductive aging. One of the most common is looking at depletion of that ovarian follicle pool. Again, mammals, female mammals are born with a finite number. So once those are depleted, the ovary does not have the ability to regenerate new eggs or new follicles. In rodents, they develop ovarian cysts as they age. In humans, primates, and rodent models, there's dysregulation of this hypothalamic pituitary gonadal axis, characterized by decreased abnormal sex steroid hormone levels and decreased inhibin. In rodents, they will have abnormal estrous cyclicity, and in primates, abnormal menstrual cyclicity. The eggs often have meiotic defects. All of these things lead to decreased fertility. And a lot of these processes are being driven by increased inflammation, oxidative stress, and apoptosis in the ovary as well as in the circulation. So what causes this early reproductive aging and senescence? There are several different factors. There's clearly a family history. We know from many epidemiologic studies that women with an early age at menopause likely had mothers with an early age at menopause. There are genetic factors, there are lifestyle factors such as diet, exercise, alcohol use. And then what I'm most interested in and what my lab studies are environmental chemicals. And we know now that there are several types of environmental chemicals, many of which are endocrine disrupting chemicals that can accelerate the onset of reproductive aging. And I'm defining endocrine disrupting chemicals as exogenous agents that interfere with the synthesis, storage, release, transport, metabolism, binding, action, or elimination of natural hormones. So essentially anything that affects any part of the hormone system. And the endocrine disrupting chemicals that we've been studying in context of reproductive aging are the phthalates. And I'm going to share with you some data from our studies on one particular phthalate called di-2-ethyl-hexyl phthalate, or DHP. And I will say that we have looked at other phthalates and mixtures of phthalates, and we see a lot of the same effects that we see with this single phthalate. So I know that many people here do environmental health and are looking at a lot of different chemicals, but I just want to give you a little background on phthalates in case you're not familiar with them. So phthalates are plasticizers and solvents that are used to make plastics sturdy yet flexible. And they're also used as solvents to dilute or to make solvent fragrances and colors. Now, because of this, phthalates are present in food and beverage containers. They're heavily in polyvinyl chloride piping, building materials in homes. They're used in children's toys, IV tubing, and medical bags. There are coatings of medications. They're used in wipes and personal care products and nail polishes, colognes and perfumes, shampoos, conditioner, and lotions. And so why are we concerned about this? Well, there are several reasons. A lot of them come from epidemiologic studies where we know now that phthalates are present virtually in 100% of people. They've been detected in lots of different human fluids and tissues. We know that men, women, and children are exposed to high levels of phthalates, but we know that the levels in children tend to be higher than in adults. And the reason for this is children are more likely to be putting plastic toys in their mouth. We also know that women tend to have higher levels of phthalates than men in general. And this is probably because women tend to use more personal care products than men. The National Health and Nutrition Examination Survey, which is a large epidemiologic study that is monitoring phthalate exposure throughout the US, has estimated now that we're exposed to over 13 different phthalates on a daily basis. Serum levels of these phthalates can be 450 nanograms per mil, and they can reach up to 250 micrograms per kilogram per day for just one phthalate. So keep in mind, we're exposed to multiple phthalates. We're also concerned about phthalates because clinical and epidemiologic studies have shown now that they're associated with an increased risk of high blood pressure, insulin resistance, decreased sex steroid hormone levels in both men and women, fertility problems in both men and women, and in women, they've been associated with an early onset of menopause. In animal models, we also know that phthalates cause adverse effects. They can affect body weight. Some of the phthalates are what we call obesogens, and they can result in increases in body weights. Other phthalates actually have the opposite problem and can result in decreased loss of body weight. We know that if phthalates exposure happens during development, they can affect development of all of the reproductive organs in male and female animal models. They disrupt the onset of puberty, usually accelerating the onset of puberty. They decrease sex steroid hormones and they reduce fertility. So one of the questions that we were really interested in is, does exposure to phthalate cause early reproductive female aging? And the studies that were out there, the epidemiologic studies at the time, really clearly showed that phthalates were associated with an early onset of menopause, but they never really went any further to look at potential mechanisms. And so we wanted to look at some mechanistic questions in humans, but we also wanted to make use of animal models to study this question in animals as well. So the hypothesis that we had was quite simple. It was that DHP exposure accelerates reproductive aging in female mice. Now, to test the hypothesis in animal models, we use adult CD1 mice, which is an outbred strain of mice, and we orally dose them for just 10 days. So I'm just going to be showing you today an acute exposure to phthalates. And we do the dosing in a gentle way where we just pipette it into the cheek pouch of the mouse. The phthalate is dissolved in our vehicle, which is corn oil, and the mice actually like it, so they swallow it. And we avoid having to do oral gavage or injections. And it's a very human relevant way of exposure because a large amount of phthalate exposure comes from oral ingestion. We were also concerned with making sure we had environmentally relevant exposures in our animal model. The 20 micrograms per kilogram and 200 micrograms per kilogram per day, those reflect the range of average daily human exposure. This 20 milligrams and 200 milligrams represents an occupational exposure or exposure that happens when people are getting IVs and having exposure through phthalates leaching out of medical bags and tubing. And then this last dose is a high dose. It's a toxicological dose that we included for two reasons. We wanted to compare our results with other studies that were done doing this dose. And we also wanted to try to get some dose response information by having a higher dose. So in these studies, what we did is we orally dosed the mice for 10 days, and then either immediately after dosing or 3 months post-dosing or 9 months post-dosing. So only dosing for 10 days, collecting samples 3 months and 9 months after dosing stop. We collected ovaries so we can count the number of follicles in the ovary because declining follicles is a marker of reproductive aging. We measured levels, We measured levels of inhibin because this is a hormone that declines with reproductive aging. We monitored estrocyclicity in the animals because estrocyclicity becomes abnormal during aging. We monitored fertility because animals, you lose fertility with age. Then we conducted RNA sequencing on the ovaries to look at a transcriptomic profile. And we conducted cytokine arrays to look at inflammatory proteins that might be changing through the aging process. Most of the data I'm going to show you are from the mice with these acute exposures. I will tell you we have done long-term exposures where we've exposed mice from the time that they are weaned throughout their adult life for up to a year now with phthalates. And a lot of the results we see also indicate increased risk of reproductive aging. There are some different things that happen with the endpoints, but I'm going to mainly show you today the acute exposure, and I'll show you a little bit of data from some of our chronic exposure. So this graph is showing the data we obtained when we were dosing for 10 days, then no more dosing, and we were looking 9 months post-dosing. And this is showing the numbers of different types of follicles in the ovary. These are primordial, primary, preantral, and antral. So female mammals are born with immature primordial follicles. These are too immature to develop an egg for ovulation, and they don't secrete steroid hormones at this point. They have to grow to this antral stage to be able to ovulate and make hormones. The important thing about primordial follicles is it's finite. If you damage those, you're not going to have any more. And if you accelerate their loss, an animal will have an early reproductive senescence. And so what I'm showing you here is our vehicle-treated animals versus our DHP-treated animals. And we see with these 3 doses of DHP a significant reduction in the number of primordial follicles compared to control. So we're causing this loss of primordial follicles that over time cannot be replaced. And then eventually these animals we suspect will run out of follicles at a much earlier time point. We start to see a little bit of a decline in primary follicles at this time point, but not enough time has elapsed to completely deplete this primordial follicle pool earlier. When we looked at estrocyclicity, the onset of irregular cyclicity, one thing we noticed is that by 9 months post-dosing, the DHP-exposed animals are already getting abnormal cyclicity, whereas the control animals are cycling normally. So mice have 4-day estrous cycles. Some of the stages are called proestrus, which we would have needed to detect for a very short period of time. So we don't have that data here. But there's estrus, metestrus, and diestrus. Metestrus and diestrus are very similar physiologically, so we most usually lump them together. So what this is showing is the percentage of days that the mice spent in each of these different estrus cycles. And we see that the DHP exposure is increasing the time they spend in estrus and decreasing the time they spend in metestrus and diestrus. So we are getting an earlier onset of abnormal estrocyclicity. I also want to point out here that phthalates are endocrine disrupting chemicals, and endocrine disrupting chemicals often have non-monotonic dose responses. So they're not linear. With more dose, you don't necessarily get more response. And if you look here, we have almost kind of a really classical response to endocrine disruptors where we've got inverted U or U-shaped dose response curves. And some of our most profound effects are not at the highest dose. When we looked at inhibin, that hormone that declines with reproductive aging, again, we saw that inhibin levels declined faster in our DHP-treated animals compared to controls. So again, this is our vehicle control and our different doses of DHP. This is 9 months after that initial exposure, and we see a decline in inhibin levels at these 2 doses compared to control. Now, ultimately, what we're really interested in is, do these animals have an early onset of infertility? And so this is 10 days of dosing, and I'm showing you one of our earliest time points. This is 3 months after dosing stopped, and we're looking at the fertility in these animals, the percent of animals that were fertile that could mate and have offspring with a control male. And what we see is that the lowest dose of DHP significantly reduced fertility compared to control. And again, we have this U-shaped dose response where the lowest dose of phthalate in this endpoint is having the most profound effect. So another point I want to make about this is you don't always get the same endpoints at the same doses. So this is why when you're studying a physiological process such as reproductive aging, you need to look at multiple endpoints. And then when we took some of these ovaries, either immediately after dosing or 3 months post-dosing, we conducted RNA sequencing analysis on them. And we analyzed the top 5% of genes that had the biggest statistical differences between control and treatment, and allocated them to KEGG pathways using David. And some of the most profound pathways that were changed were expression of genes that were tied to inflammation, oxidative stress, and apoptosis. And if you remember, I said that these kind of pathways are what are driving normal reproductive aging. And we were seeing increases in inflammatory, oxidative stress, and apoptosis pathways in response to DEHP. Now, some of these data are from cytokine arrays, where we're looking at profiles of cytokine proteins. And these are data actually from our chronic exposure study, where we're putting phthalates in the diet. And we're using 1.5 parts per million in the diet, or 1,500 parts per million in the diet as our doses. And these are showing in the ovary and the sera the different cytokine profiles that happened after DHP exposure. So at both the low dose and the higher dose, we had drastic changes in the cytokine profiles. If you look at the low dose, there were changes in 10 of the cytokines in the ovary, 16 in the sera, and 4 that were common between ovaries and sera. And at the high dose, 5 significant changes in the ovary, 12 in the serum, and 3 that overlapped. And one thing, when we just started doing these experiments, we actually expected that the cytokines would all increase, the inflammatory cytokines would all increase. And you probably notice here that some of them are significantly decreasing. Turns out there was a recent paper that came out that said during ovarian aging, sometimes these inflammatory cytokines actually decrease. So it's consistent with existing literature. We have not yet, these are brand new data where we're trying to go through and figure out what each of these cytokines does, and to follow up, maybe doing more assays to confirm this. We don't have data yet on the acute exposures, which is why I'm showing you the dietary exposures. And then this is showing you those same results a little bit differently. So this is looking at the low dose exposure and the cytokine profile in the ovary versus the high dose exposure. And again, we have some factors that are going up and some that are going down. And between low and high dose, we just had 4 cytokines that were consistently changed among the doses. And again, not sure what this means yet, whether this is going to lead to increased inflammation or not, and we're working on that now. So what I've told you so far from the mouse studies is that short-term exposure to DHP has long-term consequences on female reproduction and female aging. So when we took our healthy CD1 reproductively active mouse, we saw that DHP decreased follicle numbers, inhibin levels, decreased normal estrocyclicity, decreased fertility, and increased ovarian inflammation, oxidative stress, and apoptosis. And all of these are factors that drive the onset of early reproductive aging. So we think that this short-term exposure is having effects well after the exposure that lead to the mice having early reproductive aging. So even though my lab primarily uses mouse as a model, I think I don't probably have to say that women are not mice, and there are a lot of differences. There's a lot of similarities between the mouse reproductive system and a woman's reproductive system, but they're not identical. So what we wanted to do was translate our findings. Yes.
So, do they stay on board, or are they cleared? Are these persistent in the mice?
The phthalates?
Yeah.
Oh, so they're absolutely cleared very fast. They have a short half-life. Depending on the phthalate, the half-life is between 12 and 24 hours. So we know that once we stop dosing, and we've done some studies where we've monitored how quickly they clear it, by the time we're assessing these endpoints, phthalates are long gone.
Thank you.
Yeah. So, Okay, so what about phthalates and reproductive aging in women? So some things that are similar when you're thinking about reproductive aging in women and mice are some of the markers are very similar. So women that are reproductive aging will lose ovarian follicles. They will have abnormal cyclicity, so the menstrual cycles will become very abnormal. And in women that have not had a menstrual period for 1 year, clinicians will define them as being postmenopausal. So they've transitioned from the reproductive state to a postmenopausal state. Women also have abnormal hormone levels that are characterized by low anti-Müllerian hormones, low inhibin, and low levels of sex steroid hormones, estrogens, progesterones, and androgens. Women will also have the onset of menopausal symptoms. And the most common one in women is hot flashes. That's a big difference between mice and women. Women get hot flashes, mice do not have something. And aging in women's ovaries is also driven by oxidative stress. And in women, we can measure some of these markers for ovarian oxidative stress or systemic oxidative stress by measuring 8-hydroxy-2'-deoxyguanosine oxidative damage or total antioxidant capacity. Now I've put in red here the things that we've looked at in a human study that I'm going to share some data on sex steroid hormone levels, hot flashes, and these other markers. We're still in the process of analyzing some of these other things. So to give a little background on what the decline in sex steroid hormones looks like in women as they age, this is a schematic showing women from age 25 to 75. If you look at estrogen here first, it's pretty consistent until the late 40s, early 50s. There's this dramatic decline with a lot of fluctuation in that decline, and then eventually low levels that are stable. Progesterone is a hormone that also declines with aging, but it tends to decline around the same rate as estrogen, but that decline starts much earlier in women than the decline in estrogen. And then testosterone levels also decline in women, but that decline is much more gradual. Now, hot flashes, which is another marker that I'm going to talk about, I want to give a little background on this. Hot flashes are the most common menopausal symptom. This is actually why women going through menopause go to their doctors, because they want relief from these hot flashes. They're characterized as sudden periods of intense heat in the upper body, arms, and face, and they're followed by flushing of the skin and profuse sweating. They sometimes can be followed by chills, palpitation, and heightened anxiety. So more than 1 million women in the US each year are entering menopause, and between 50 and 70% of them are experiencing hot flashes. And this might sound like a trivial thing, but for women that have them, it's very uncomfortable, and it's a quality of life issue, and it drives them to seek healthcare. And in fact, hot flashes alone result in about $340 million a year in medical expenses. So it's quite serious. From a public health standpoint, hot flashes are thought to be indicative of other serious conditions that might come on later throughout the menopausal transition. And these include osteoporosis, cardiovascular disease, depression, and early death. The reason for this is that women that are having hot flashes, those are being driven by those declining levels of estrogen. And heart disease, osteoporosis, and depression are all driven by low or changing levels of estrogen. And because these women have an increased risk of heart disease, osteoporosis, and depression, all of those collectively can lead to conditions that result in early death. So it's thought that hot flashes might be a really early indicator of problems with reproductive aging to come. And then lastly, I just want to give a little bit of background on oxidative stress during reproductive aging in women. So to define what I mean by oxidative stress, it's an imbalance between the production and accumulation of reactive oxygen species, which can then target DNA, protein, and damage them, and antioxidants. So what happens is the body starts to make more reactive oxygen species that cause damage, and it has less antioxidant capacity. Now, we know from many studies that oxidative stress occurs naturally with aging and that it increases during menopausal transition. And we know from lots of animal studies and some human studies that phthalates increase the risk of oxidative stress. We wanted to use oxidative stress markers because a study I'm going to tell you about, we had a lot of stored serum samples from women going through the menopausal transition, and we can easily measure this 8-hydroxydeoxyguanosine oxidative DNA damage, so a factor that leads to damage from oxidative stress, and total antioxidant capacity. The hypothesis for these human studies is that phthalate exposure is associated with low sex steroid hormones, hot flashes, and oxidative stress in midlife women. So to test this hypothesis, we made use of a cohort study that I started a long time ago when I was at the University of Maryland. And it's a prospective cohort of midlife women. We enrolled 782 women in the study. And at the time we started this study, we were really interested in risk factors for hot flashes. So we required that women be either having or not having hot flashes at baseline of the study. The women in the study were followed between 1 and 7 years, depending on the date of enrollment in the study. And the average follow-up was for 4.5 years. So every woman in our study came to our clinical site between 1 and 4 times a year, each year that they were in the study. They completed detailed questionnaires that assess reproductive history, hot flash history, demographics, lifestyle history. We weighed and measured the women so we can calculate BMI, body mass index, and all of the women donated blood and urine samples. Now, the study population was recruited when I was in University of Maryland, which is in Baltimore City. And I had a clinical site at Johns Hopkins University and their Department of OBGYN. And so here is where Johns Hopkins and University of Maryland are. All of these light blue areas are where women were recruited and participated in the study. And we really focused on these areas because they were within driving distance of the clinic site. So to be in the study, as I said, women at baseline either had to be having hot flashes or not having hot flashes, but they all had to be between 45 and 54 years of age. The reason we had this criterion is because average age of menopause in women is around 50 to 51. So we wanted to catch women before they were entering the menopausal transition and follow them through it. And NIH only gives grants in 5-year increments, so we couldn't recruit really young women and then wait a long time. We wanted the women to have intact ovaries and uterus so that we would capture women naturally going through menopause. If you remove the ovaries and the uterus, a woman's going to have a surgical menopause. We wanted the women to have at least 3 menstrual periods in the last 12 months so that we could make sure we were not capturing postmenopausal women. So they had to still be cycling or even starting in perimenopausal where you start to get those erratic ones. The women could not be pregnant and they could not be taking hormone replacement therapy, herbal supplements, or hormonal contraception. And the reason for this is our initial study was to look at risk factors for hot flashes. And all of these are treatment for hot flashes. So we didn't want women being treated for hot flashes. And they couldn't have a history of cancer. And the reason for this is many chemotherapeutic agents target the gonads and they kill ovarian follicles. And so we didn't want women that were having menopause because of their cancer. And obviously, if a woman has cancer, she needs to be treated for that. That's the biggest priority. So in our study design, the women completed these questionnaires that assess demographics, details on hot flash history, risk factors for hot flashes and menopausal symptoms, and confounding variables. And some of the data I'm going to show you, we used that in our data analysis. Then from all of the women at every clinic visit, we collected serum, and we used that serum to measure sex steroid hormone levels and to measure our 2 markers of oxidative stress, this AOHGG, which detects oxidative damage, and total antioxidant capacity, which measures how capable a woman is of preventing or dealing with reactive oxygen species. From the urine, we measured urinary phthalate metabolites. The reason we did this is because phthalates have a very short half-life, and the gold standard in epidemiology studies is to measure the urinary metabolites as a surrogate for how much exposure a woman might have had. So for the marker of hot flashes, we had some very specific definitions, and I'm going to show you some data related to this. So we defined women as having any hot flashes if they reported on their questionnaire experiencing them around the time they were approaching menopause. Self-report of hot flashes is considered the gold standard for reporting this. It's actually the most accurate way to do in a large population-based study. We also measured severity of hot flashes. If they were mild, they would have a sensation of heat. Moderate was described as sensation of heat with sweating, and severe sensation of heat with sweating that disrupted usual activity. And we monitored the frequency of the hot flashes, whether they were occurring daily and weekly or monthly. We did have some women in our study that had a hot flash every 3 months or every 4 months, but a lot of women that were having them quite frequently. Now, for the urinary phthalate metabolite assessment, we based our assessments on a large body of epidemiologic studies where they use summary measures to try to capture exposure from different types of phthalate exposures. So one of these is what we call sum of personal care products. This sums up the urinary metabolites that would most likely be coming from personal care products and is a surrogate for exposure of phthalates from personal care products. We have summary of DHP exposure, which we summarized the metabolites coming from DHP, a summary measure of anti-androgenic metabolites, which summarizes those that have known anti-androgenic activity. We had a summary measure where we looked at exposure, surrogate measure where we looked at phthalate metabolites that would come from plastics. And then we had a summary measure that just had all the phthalate metabolites. So just to give you a little bit of information on the study, we had at baseline 332 women with hot flashes and 400 women without hot flashes. The study, all of the women had to be between 45 and 54. 53% of the women that had hot flashes were 45 to 49, and 46% of the women that had hot flashes were 50 to 54. In the no hot flashes group, the majority of those were 45 to 49, and there was a significant difference with younger women being more likely to have no hot flashes. And this kind of makes sense if you think about it. They haven't started to transition through the menopausal period yet, so they haven't started experiencing hot flashes. With education, about 43% of our women had some college, tech school or less and had hot flashes, and 56% had hot flashes and graduated college or more. And here we saw a significant difference in education level and risk of hot flashes. The majority of women that did not have hot flashes tended to have graduated college or more. We have no idea why higher education reduces the risk of hot flashes. It may have to do with maybe they didn't live in old homes that were having some chemicals that would be causing hot flashes, or better diets, etc., but we don't know. We did not see any racial, ethnic differences in risk of hot flashes or in employment status. So for the phthalate summary measures, I'm showing you here what the phthalate metabolite levels look like in the urine from these women. So these are the different summary measures I explained, and this is their abbreviation here. This is in the study, our cohort study, the Midlife Women's Health Study. And I'm showing the median and 25 and 75th percentile levels of these different phthalate metabolite summary measures. And I want to point out here, these are our measures, and I'm comparing them here to women in the Ed Haynes study, this nationwide study across the United States that are in the same age range as our women. And we have a lot of congruence or a lot of similarities. So we feel like the women in our Baltimore study are at least fairly represented as far as phthalate metabolites as to the whole US population. So what I'm showing you here is some of the data where we were looking at phthalate exposure and how estrogen, testosterone, and progesterone levels changed over the time, over the cohort length of time of follow-up. And what I want to point out here is this is showing increasing levels of phthalate exposure based on quartiles. And what we're seeing is that as phthalate exposure is increasing, that's resulting in, over time, a decrease in estradiol in these women. We also expected to see a decline in testosterone, particularly because a lot of male studies, epidemiologic studies, show quite nicely that phthalate exposure is associated with decreased testosterone levels. But we did not see any significant changes in testosterone. And actually, we're quite surprised to see progesterone. We expected that also to go down. It goes up with phthalate exposure. And I have no idea what that means. So I would love to hear from people if you have any hypotheses. And we've looked at this a million different ways. So I'm convinced this is what the data look like. And I do think there is something different about phthalate exposure and its association with hormone levels in midlife women as compared to young women, pregnant women, and compared to men. So there's something about this period of the lifespan where we have some different effects going on with hormones. Now, when we looked at the aging marker of hot flashes, I'm showing you here in the white circles, premenopausal women in the study and perimenopausal women. And then if you look first at panel A, this is showing whether the women experienced hot flashes in the last 30 days, so recent hot flashes, and looking at the odds of experiencing hot flashes in the past 30 days for every twofold increase in phthalate exposure. And so the way I look at these is if you look at the 1 here and this line here, if the value falls on that line or overlaps with that line, it's not a significant difference. If it falls to this side of the line, there's an increase in risk, and this side of the line, there's a decrease in risk. So when just looking at recent hot flashes, we saw one association with the metabolite summary measure of those in plastics in premenopausal women. However, when we looked at the frequency of hot flashes as characterized by daily and weekly, we see that the summary measure of DHP, plastics, and all of our phthalates increased the risk of frequent hot flashes compared to women not having hot flashes or having monthly hot flashes. So it seems like phthalate exposure may not be associated with whether a woman ever gets hot flashes, but among those women who get hot flashes, it's associated with an increased frequency of hot flashes, which is actually a big concern because it's those frequent hot flashes that affect quality of life and bring women to the doctor to get relief from it. And then looking at our oxidative stress measures, this is 8-OHDG, which detects oxidative damage. And here again, I'm showing the summary measures of phthalates, but I'm also showing you individual data from some of the different phthalate metabolites. And what this is showing is that a lot of the individual phthalates and the summary measure, particularly for some of DHP, significantly increased the presence of this marker of oxidative damage. We have this one weird phthalate here, MCPP, that significantly decreased this oxidative stress marker. Again, I have no idea why this is happening. I've tried to look to find more and more about this particular phthalate, and I don't see anything that gives me a clue to why this is happening. So again, I'd love to hear some suggestions if you have them. And then when we looked at the ability to have total antioxidant capacity, so having looking at the phthalate exposures, individual phthalates, as well as the summary measure, we see that a lot of the phthalates decrease total antioxidant capacity. But again, here's MCPP, which has the total opposite effect as some of these other phthalates. And some of the summary measures and some of the individual phthalates were not significantly different. So what I've told you from this midlife women's health study is we're starting to think that phthalate exposure might be associated with early menopause because it's associated with some markers of reproductive aging in women. And we're in the very early stages of this study, but what I've shown you so far is that we know phthalate exposure decreases estrogen levels over time, increases the frequency of hot flashes, and increases risk of oxidative stress because we're reducing antioxidant capacity and increasing oxidative damage. So we think that phthalate exposure is associated with reproductive aging in women, but we still have a lot more work here to do. So to conclude, I hope that I've shown you, or at least convinced you, that phthalate exposure may be associated with early reproductive aging in female mice and midlife women. In mice, we think that this is really, we're pretty confident about this because phthalate exposures decrease follicle numbers, inhibin levels, estrocyclicity, and fertility, while increasing inflammation, oxidative stress, and apoptosis in the ovary. And in the midlife women's health study, we see an association between phthalate exposure and low estradiol levels and increased risk of oxidative stress and hot flashes. So before I end, I just would like to thank my wonderful laboratory at Illinois, particularly Zane Inman, who's my PhD student, who's brave enough to do both animal studies and learn enough epidemiology studies to participate in that. And so much of what I showed you is a lot of his data. I'd also like to thank Lori Reitzman and Romana Novak at Illinois, who are my collaborators on the mouse studies, Serdar Balan, Ping Yang, Rita Strakowski, Russ Hauser, Tamara James Todd, and Ryan Babaldi, who are all collaborators on the human part of the study. Rita and Russ, Tamara and Ryan are all proper epidemiologists, so they are helping us with all of this. And then I'd like to thank Howard Zacher from Johns Hopkins, who is an OBGYN that helped us with all the recruitment and obtaining the samples, and Raj Kumar at Colorado State, who helped us analyze some of the gonadotropin data that we showed. So, oh, and thank the funding. And I will thank all of you for your attention, and I'm happy to answer any questions.
I've got one in the chat. We have a question in the chat from Dr. Yucheng Liu. He wants to know, I think, about the cohort study. Why did you classify participants into two groups based on hot flash status? And also a second question, how do you distinguish the increase in oxidative stress caused by aging versus by exposure?
Yeah, so great questions. So I'm only showing you the data where we broke it out by hot flashes status because the initial intent of the parent study was to look at risk factors for hot flashes. So that's why we were doing that. But now that we're doing more analysis stuff, we're not going to just break them out by hot flashes status. We're actually going to be looking at some of these biomarkers and how they change. And a lot of what I've been showing you is that kind of at baseline, and we have longitudinal data on these women. So what we want to try to do is say, okay, we had phthalate exposure in this clinic visit. How does that correlate with what hormone levels looked at in the next visit? So we can have things in a forward longitudinal way. And then the second question, I forgot.
By the same person, how do you distinguish the increase in oxidative stress caused by aging versus by exposure?
Yeah, so really hard to do in a human study, right? So I don't know, I don't have a good answer for that. In the animal studies, we can do it because we can do in vitro studies where we culture ovaries in dish from young and old animals, and we can see that we've got increased oxidative stress and inflammation, even in a younger ovary that's not been through aging. But I think it's harder to do in a human study because women are not going to give up their ovaries for this study. Okay, so.
I got one more in the chat if nobody has one right now. Kathy Peltz asks, is phthalate exposure cumulative throughout life or can it be cleared?
So it's got a very short half-life. So most phthalates less than 24 hours. So it does get cleared, but we're constantly exposed. And so we always have measurable levels of phthalates. So can't be completely cleared.
For your phthalate stuff, I thought it was a lovely presentation. Thank you so much. I was wondering, because you were showing how there's like midlife and how the phthalate exposure might not indicate whether or not you get a hot flash, but the frequency and maybe intensity of them. Do you have any thoughts on whether or not like having higher levels of phthalate exposure, like in your 20s, early 20s might indicate, or do you think it's something that happens, like a tolerance is like reached in the 30s where that early part hits, I guess is my question, if that makes sense.
That's a great question. I don't know. Actually, I do know that there have been studies done a lot of times in women undergoing IVF or in young populations of women. And there is an association between phthalate exposure and loss of follicles or low estrogen levels or increased infertility. But I don't know of a study where they've followed women from age 20, looked at their phthalate exposure, and then looked at what happens when they're having hot flashes much longer in life. I can tell you from some animal studies that we've done that a phthalate exposure in early life, depending on the phthalate, the dose, all of that, can result in outcomes that we see later in life that aren't hot flashes, because mice don't really get them, but are things that would lead you to believe that that could happen.
Thank you for visiting. So I got a human question and then I'll ask a big mechanistic kind of question. So in your demographic table, you did stratify by like 40 to 50 or whatever. What happens if you stratify for your results? So your estrogen, your progesterone, and your hot flashes by
Yeah.
those age groups?
So yeah, so we've done this, so I have to think this through. So it's in the, with the hot flash, so if we stratify them by age, we do see that the older women will be reporting hot flashes compared to the younger women because they are getting towards that age where they're starting to have them. Does that make sense? With the, you asked me about age.
And then the hormones.
And the hormones. So yeah, so we've looked at like how hormones change longitudinally, and they decline. If you do it by age, we see like a nice age-related decline in all of them. What we think, like with the progesterone, one of the things that we've been thinking about is, so in a normal healthy woman, progesterone is one of the first hormones to decline, but in our study, we saw like an inverse thing of what we expected. So we're wondering if maybe the age range of women that we're capturing, we missed that decline in progesterone and we're getting some kind of secondary progesterone from somewhere else later. So we didn't have women young enough to be able to like go back in time to look at, because 45 to 54 is a pretty narrow age range. So like even looking at things by age and hormone level, we'll see the decline that happens in each woman with her age, but we don't see like this dramatic shift.
And then you kind of briefly talked about this, but a little bit more about when your mechanistic endpoints don't line up with your phenotypes. Can you talk a little bit about why that might be specific to phthalates or not specific to phthalates?
Yeah, so I feel like this might be true, and I don't know, people that work on other EDCs can tell me this. So really the effect you get from things like phthalates, it depends on so many things. It depends on what tissue you're looking at, what endpoint you're looking at, what dose and what kind of phthalates. So we don't always see, like, for example, I would have thought at one particular dose of phthalate, we should see decreased follicle numbers, decreased estrogen, decreased fertility, like all of those should be down, but that's not the case. And I think it's probably because different phthalates affect different tissues in different ways. So for example, the decline in follicle numbers, that's going to be driven by usually a direct effect of the phthalate in the ovary, but it could also be affecting the hypothalamus and the pituitary, which then cause an effect on the ovary, right? So sometimes you can see an effect like in the ovary that might not be driven by the effect of the phthalate in the ovary, but it's affected another tissue. And that other tissue might have its own phenotype as well. I'm trying to say, so you can direct and indirect effects depending on whether it's hitting the brain, the pituitary, the ovary, whether it's in the blood, how it gets metabolized. So the ovary in some ways is a big target because it can convert DHP to one of its more toxic metabolites, MEHP, right in the ovary. So some of the effects we're getting in the ovary that we don't see in other tissues are probably because the ovary is converting DHP to MEHP, whereas that's not happening in other places.
Complicated.
Yeah,
And
it's
hard to
way.
find a mechanism to kind of intervene at if all
Yeah.
these different things are happening.
I initially thought we would get the same mechanism, but that's not the case. And it became even more complicated when we started doing transgenerational studies. And boy, you don't even get the same phenotype in all 3 generations. Oh, yes, go ahead.
So you mentioned herbal supplements. Do you know offhand which ones are used, and is there a mechanism of action that's shared?
Yeah, so there was, and this is going back quite a few years, there was a lot of women that were taking herbal supplements that were basically like soy products like genistein, and those things. And there have been, that kind of came out of the fact that there were epidemiologic studies showing that Asian women that ate soy-based diets didn't have hot flashes as much. But I think subsequently, we now think that maybe it's they don't report them as much. It's a cultural thing, so they're not talking about them like American women do. And there have been some studies specifically looking at those supplements and whether they're effective in alleviating hot flashes, and they're all over the maps. Some say yes, some say no, some say they make them worse. And so we just said we're not going to have that as a confounder in our study.
Another one from Dr. Liu. Have you done any multivariate analysis? And also he says, thank you for conducting both animal and epi studies to test your hypothesis. Wonderful work.
Oh, thank you. So, yes, we have done some, but and by we, I mean Rita Strakowski, and not me, because I am not anywhere skilled to do that. But we're just starting to do that now, to look at multivariate analysis and also to do some mixture type analyses to look at that more carefully. So we're doing it, but haven't done a lot of it yet.
All right, let's
Thank
thank Jodi again. Thank you.